Welcome to This Explainer. Today, we're doing something a little different. We're going to act as biological architects to break down the highly technical, beautifully complex process of gene cloning. But we're not just going to read through the science, we're going to visualize it as a clear, step-by-step factory blueprint. We'll explore exactly how modern biotechnology takes a single microscopic instruction and turns an entire cell into a manufacturing powerhouse. Let's start with a massive number to set the stage. 3.2 billion. That's roughly the number of base pairs in a single haploid human genome. It's an absolutely staggering amount of genetic information, all packed into the microscopic nucleus of a cell. Now picture this. You want to find and clone just one specific gene, right? Maybe it's about 3,000 base pairs long. Compared to that 3.2 billion base pair total, it is literally harder than finding a needle in a haystack. It's like 1/1,000,000th of the genome. So obviously, that's a massive biological hurdle we have to solve right out of the gate before any cloning can happen. So here's our game plan for today. We're breaking this down into 5 major phases. We'll start by drafting our blueprint by finding the candidate gene, then isolating the nucleic acids, cutting and copying the DNA, delivering it to host cells, and finally, running some strict quality control with blotting techniques. All right, phase 1, finding the candidate gene. Because, we can't build a factory if we don't know exactly what biological instruction we're trying to manufacture. We pick these candidate genes completely based on their real-world value. Are we trying to cure a disease? Well, then we might hunt for the gene that produces insulin to treat diabetes. Are we looking to boost agriculture? Maybe we need a gene that fixes iron deficiency in plants, which is known as chlorosis, or one that gives crops salinity tolerance so they can survive in really harsh environments. We even look for genes that encode the spike proteins of viruses, which, as you might know, is the exact blueprint used to design modern mRNA vaccines. Once we spot that value, boom, we lock it in as our target. Moving right along to phase 2, isolating the nucleic acids. Now that we have our target, we actually need to go in and gather the raw genetic materials from the host organism. We need to extract that DNA. This isolation process is just a beautifully systematic chemical extraction. First up, we physically rupture the cell membrane to spill everything out. But cells are packed with enzymes that love to chew up loose DNA, so step 2 is quickly adding buffers to protect our nucleic acids. Next, we mix in chemicals, specifically phenol and chloroform, and this separates the proteins and lipids into an organic phase, leaving our precious nucleic acids floating safely in a watery aqueous phase. Finally, we pipette that water layer out, drop in some chilled ethanol, and the pure DNA literally clumps together. It precipitates right out of the liquid so we can easily spool it up. Which brings us to phase 3, cutting and copying DNA. We've got our pure, raw DNA isolated in a test tube. Now it's time to break out the biological toolkit. To cut the DNA, we use cellular scissors called restriction endonucleases. Now, out of the 3 main types of these enzymes, Type 2, like ECO-RI and BAM-HI, are incredibly special. They have entirely separate endonuclease and methylase functions, and they cut specifically within their recognition site. Types 1 and 3, they kind of just cut randomly at a distance. So that absolute precision makes Type 2 the gold standard for genetic engineering. They let us snip the DNA exactly where we want it. But here's the catch. Once we've successfully snipped out that tiny 3,000 base pair gene from the massive 3.2 billion base pair haystack, we still only have one microscopic copy. So how in the world do we manufacture enough of this blueprint to actually build our factory? Enter the biological photocopier, PCR, or polymerase chain reaction. It's basically an incredibly precise cycle of temperature changes. First, denaturation hits around 95 degrees Celsius, melting the double-stranded DNA into 2 single strands. Second, we cool it down to about 50 to 55 degrees for annealing, which lets specialized markers called primers latch onto our target sequences. Third is extension at 72 degrees. This triggers a heat-stable enzyme called DNA polymerase to race down the strand, adding base pairs and building a brand new double helix. And the math behind this PCR process was just wild. Every single time you run through those 3 temperature steps, the number of DNA copies, which we call amplicons, doubles. It's an exponential curve, 2 to the power of n. Cycle 1 gives you 2 molecules, cycle 2 gives you 4, then 8, and so on. If you repeat this for just 25 to 30 cycles, you literally turn a single strand of DNA into millions or even billions of perfect copies in just a few hours. Okay, phase 4, delivering DNA to cells. We've got our instructions, and thanks to PCR, we have millions of copies. Now we have to actually ship them onto the factory floor. We need to get this DNA inside a living host cell. Getting inside is tough because the cell membrane is heavily guarded and naturally repels foreign DNA. So, scientists have engineered several truly fascinating biological delivery trucks to outsmart the cell's defenses and force our genetic payload inside. We actually have a whole arsenal of techniques for this. We can use chemical transfer, bathing the cell in calcium chloride and then shocking it with 42-degree heat to force its pores open, or electroporation, where we zap it with rapid pulses of electricity to punch microscopic holes in the membrane. Then there's liposuction, where we basically disguise the DNA inside hollow fat bubbles that naturally fuse right into the cell. For bigger cells, we can literally just push it in with a tiny needle using microinjection. And my personal favorite, the BioListic method. We literally coat microscopic gold particles with our DNA and blast them directly into the cell using a gene gun. All right, finally, we arrive at phase 5, screening with blotting techniques. Our host cell has accepted the DNA, and our biological factory should be up and running. But we need strict quality control to prove it actually worked. To verify our success, we use specific screening techniques called blotting. And there's a crucial distinction here. We use northern blotting when we want to check if the new DNA is successfully transcribing RNA. Basically, are the factory workers actually reading the blueprint? But we use western blotting to identify if the final desired proteins are actually being built. So remember, RNA for northern, protein for western. The workflow for this quality control is super elegant. First, we use an electrical field to push the cell's molecules through a gel which separates them strictly by size. Second, we electrotransfer those separated molecules onto a sturdy nylon membrane. Third, we bathe that membrane in custom-designed labeled probes that will only stick or hybridize to our specific target, RNA or protein. Finally, we expose it using autoradiography. If our factory is working, those specific bands will light up like glowing barcodes on an X-ray film, confirming absolute success. So there you have it, from searching a billion-letter haystack to isolating pure DNA, copying it exponentially with PCR, blasting it into a cell, and proving it all works with glowing barcodes. Whether it's synthesizing critical insulin or deploying life-saving mRNA vaccines, this instruction manual is just the foundation. The tools are clearly defined and ready to go. The only question now is, what biological factory are we going to build next?